let's see this. Okay, so the first part of this is going to be an introduction to Raman spectroscopy, and I emailed you those slides. Our exam got moved from today to Tuesday, November 24th. It will predominantly be proton, carbon, and 2D NMR, okay? There will be no other nuclei on it. There will be no Raman on it, okay? So that's where I want to focus our attention on that. And so I'll give you the empirical formula, proton, carbon, and then we'll do a couple exercises where figure the whole thing out. You know, here's the IR, here's the molecular formula, and here's some things figured out. But then there'll be some exercises like we did last time where it's predict the 2D NMR, assign all the different protons and carbons in the 2D NMR. That's what we're going to do. Yes, is there a question? No? Okay.
So there will be only 2D NMR?
Proton, carbon, and 2D NMR.
Okay.
Yeah.
And what about the UV and what about the UV?
There will be one problem that's solving everything. You know, so you'll get an IR and UV is not going to be on there. Actually, no, I need to do, we'll probably have, I'm sorry, UV is on there. I forgot, UV is not on my schedule here. There will be one set of questions on UV, including predicting the lambda max. Okay, so yes, thank you for reminding me of that. I have it on there, I just forgot I had it on there.
Yeah, you previously told that there will be a question for the UV.
Yeah, it's like a 2 or 3 part question on UV. Yeah. So thank you for reminding me. Okay. So let's go ahead and I'm going to pull up my slides and we're going to start talking about Raman spectroscopy. Now, what's cool is that I've done this in the past at Texas State, which is how I knew there was an instrument over in physics, but I also published on a certain facet of it called surface enhanced Raman spectroscopy, or SERS. And I'll talk to you about that as well. Let me go ahead and do my screen. So as if you've had a chance to be exposed to Raman spectroscopy, you'll see that it actually complements IR because they are basically looking at the same frequency section, and a band in Raman will directly correlate to a band in the IR as far as functional groups and the types of groups we see. Okay, so those bands do correlate. However, Raman spectroscopy takes into account the things that IR doesn't. And so let's go ahead and look at the compare and contrast. Okay, so first and foremost, in Raman spectroscopy, we are looking for a change in polarizability, not a change in dipole. So that's the big difference between them, okay? So in IR spectroscopy, we typically look at the absorption of an energy. In Raman spectroscopy, we're actually looking for an emission and whether or not that emission has a change in frequency. So this is it's different in how we do the thing, but it's interesting that the two systems complement each other and represent the same frequencies and the same functional groups. So to compare and contrast, IR is all about vibrational modes, and it's the absorption of that IR energy making those modes vibrate. In Raman, we're also talking about those same vibrational modes. However, we aren't talking about having an unsymmetrical or any kind of vibration that changes the dipole moment is what we call IR active, okay? So these asymmetric vibrations are IR active because you have changed the dipole and we see them in IR, okay? What if you have some kind of vibration where you don't change the dipole? For example, let's say you had a carbon-carbon triple bond and it doesn't excite very easily in IR, but if it has two of the atoms moving in the same direction or the other direction, they're moving in different, opposite each other, but not the same magnitude, we now have a change in dipole. What if those two carbon atoms were extending and then compressing and extending and compressing and not changing the dipole? all we've done is changed the momentary distribution of the electrons around the outside of the system, elongating those electrons, going back to our neutral state and compressing our electrons. Okay, so in this case here, we haven't changed the dipole and we don't see it in IR, but we do see it in Raman. So that's one of the interesting things about the Raman spectroscopy. We're looking for, it works a lot on symmetrical or cylindrical type of atoms. I'm sorry, types of bonds and molecules, because in those systems you can have this equal and opposite vibration mode, which is not IR active. Okay, so that means that we have certain, even though it's the same functional group, there are certain vibrations that are, one is going to we see in IR and one we're going to only see in the Raman. Okay, so the idea here is that we have to excite it with some kind of energy, and usually we excite in Raman spectroscopy with light. Okay, so when light is absorbed by the system, it can do a couple different things. Okay, it can not be absorbed, go straight through, or it can have an interaction with the electronic shell of the atom and or molecule, and it can do what we have where the energy is absorbed and then the energy is released, and they're the same amount of energy. And so we call that Rayleigh scattering, meaning that it is interacting with the system but not being absorbed and being released at the exact same energy. Okay, now in Raman spectroscopy, we're actually looking for that same absorption, that same scattering out of the angle of the directed light, but we're looking for a change in the emitted light. Okay, when there's a change in the emitted light, that means that we've gone to different energy states, and that gives us our, number one, our energy is the change in that, and number two, it gives us two different types of transitions to look for. Okay, so in this right here, we have our lowest electronic states. And those, I mean, our ground states are the ones where the electrons actually reside. They're the real orbitals and electronic states where the electrons are. Then everything else is our LUMO set, our set of infinite number of excited states that don't exist until you excite an electron in them. So those are those, you know, semi-infinite number of places to put electrons in an excited state. Okay.
So what is, sorry, I have a question. So what is the difference between the UV sort of thing and this one? Like it's look familiar to their transition sort of thing.
Again, UV is an absorption. We're looking at absorption. IR, we're looking at absorption. So we're looking for the lack of that wavelength in our spectrum. Here, we're actually going to be measuring the light coming out of, being scattered from the system. And that's going to be what we're measuring. Okay, so that's the big difference. So imagine that we have to hit it with a lot of light, because a lot of these transitions are not necessarily easy to do. And then we have to have the light actually scatter in a direction where the detector might be. We can't put the detector at 100 at the same incident beam because it'll be overwhelmed by the light it has. So it has to be off to the side. And then we're only looking at the light being scattered and its change in frequency. And that is what we're looking for there. Okay.
Okay, so the reason for scattering is the electron distribution.
Yeah. Well, in UV, again, that we have that same thing happen in UV where energy is being absorbed. But in the UV absorption, we're looking at that is the amount of energy it takes to change that electron to that specific HOMO or LUMO state. Okay. For example, going from a pi state to a pi star state is a specific energy, and that specific energy is the wavelength of UV light we use. In Raman spectroscopy, most of the time we use visible light as the excitation, and we have to use a very intense visible light for that excitation. So we typically use lasers. Okay, so that's not the energy that would make the molecule vibrate. That's not the energy that would make an absorption in the UV. This is a lower energy. And so we're having not to look at those states. We're looking at when it does do an excitation, what is the change in frequencies given off at 90 degrees from the incident light. Okay, so that's why it's different. Oh, you're being taught an instrument about Stokes and anti-Stokes calculation? Good, I'm going to talk about that in a little bit. Okay, so it's all about the change in the energy of the incident light. Okay, so let's look at that again. Okay, so if there is no change in energy from the scattered light coming from the system, that means that you had the same amount of energy got to the excited state as was emitted during the scatter. Okay, and we call that elastic scattering because it's one-to-one energy wise. Okay, now if you have the emission from the system, the scattered light from the system, having a different energy than the excitation energy, then it can be higher or lower. And we call that inelastic because it's not exactly one-to-one, it's one minus something or one plus something. Okay, and that difference in energy comes from that difference in which energy band they're relaxing back to during that emission. Okay, so elastic is the Rayleigh scattering, and that's okay. But Raman is the one we can actually measure these different functional groups with in that IR frequency band. Okay, so now let's talk about Stokes and anti-Stokes. Okay, so let's look at this graph here. And so let's just say we have an excited state going to a ground state going to one excited state. And then we have another bit of energy that started from the second ground state and went up to some other energy. Okay, so in a Stokes type system, we would see the excitation electron going from this, and it's not a single electron, it's the system of electrons. So it's not quantized, it's not just an individual electron, it's the entire system working together. And so we have, if the emission goes from that first excited state back to that same ground state, we have no change in frequency, and we have that elastic Rayleigh scattering. And the same thing would happen with your second not quantized, your second excited one. If it goes back down to the exact same state, we again have no change in energy, and therefore it is Rayleigh scattering. Okay. Now, in contrast, in Stokes, we have a going from our highest occupied energy, that electron started in the number one ground state, but it's going down to the zero ground state, meaning that it now has a greater amount of energy than the energy it took to excite it from the ground one state to that excited state, because we've now drowned all the way past the one and it gained energy. And we call this the anti-Stokes mode because we actually gained energy in that emitted light. So it shifted to a higher frequency. Okay. Now, in the case of our ground state, zero state, going up to our first excited state, if that energy is emitted and goes not back to that zero state, but goes to that one state, that means the emission has less energy by whatever difference in energy between the zero state and the one state was, and therefore it is lower in frequency, the emission is, than the excited state would have been. And we call that a Stokes shift. Okay. So in the case of this, we have two possibilities here, but statistics is running against us in being able to see these things. Okay. So if we think about that, our excitation from our lowest state to our upper, and then going back down, but not to its lowest state, to a state above its lowest state, gives us our Stokes shift. Okay, because it has lower energy, we didn't emit that last little energy burst here, and therefore we have a lower energy and a lower frequency, lower frequency, yeah, sorry. In the case of an elastic one, this would be our Rayleigh shift right here. It didn't change. The excitation in, excitation out, the scattered frequency is exactly the same. And it's a very strong peak because that is more common type of decay of these excited states. Now, in the case of having our electron excited from our one state up to some level and then emitting light on its way down to the zero state, we've added energy to the system. And so we end up getting to a higher energy state in a higher frequency state. Now, the problem is that the probability of these emissions are very, very low. Okay, Rayleigh is much, much stronger than our Stokes state, and our Stokes state is actually much, much stronger than our anti-Stokes state. So what that means is that we have to use very high intensity impinging light to be able to see that light shift up and down. So let's say we're hitting it with 500 nanometer light. And so what we're hoping to see scattered out of it is something that's a little bit below 500 nanometers, giving us our Stokes energy shift. If it emits at 500, if the light we're seeing out the side is a 500 nanometer light, then that's a Rayleigh scattering and doesn't tell us anything. But if it emits at a higher frequency, then that's our anti-Stokes shift. Okay, so one of the problems with that is that means that the efficiency of excitation versus that scatter we see is actually really low. It's 0.001%. Okay, so what does that mean? If it's so low, that means we have to hit it with very high energy light. And most of the time we do this with lasers, specifically visible lasers. Number one, for safety. Number two is that we can actually use our spectrophotometers to measure the difference in emitted light, and we can get to our calculated Raman spectrum. Okay, questions on that, on Stokes shift and anti-Stokes shift? So the most of the time when we actually analyze a Raman data, we're actually looking at our Stokes shift in our translated Stokes shift emission that we have there. Okay, all right.
Like I have a question. Sorry.
Okay.
So the formula is E is equal to HV. So higher energy should have the higher frequency. Sorry.
Higher energy should have higher frequency.
So why the anti-stroke is showing less and the stroke is showing greater?
This is it doesn't have, we don't know which direction that axis is going.
Okay.
So imagine this, so frequency is increasing in this direction for this. So this is frequency here. And so we had our initial frequency plus our frequency difference here. So it should be increasing frequency in the going right. So this Stokes is higher in frequency.
And we don't know what is on the y-axis?
The y-axis is relative intensity.
Okay.
Almost always they're done in relative intensity.
Okay.
Okay.
I was also curious, what are the subscripts EX and V referring to?
The V is frequency. EX is the excitation frequency. So I said if we emit as blue light, I mean, if we're exciting with blue light right here, the scatter we get from it is going to be blue light minus some frequency. And that frequency is related to that difference in energy between the ground 1 state and the ground 0 state. Now, again, this is the frequency of the excited light plus the extra energy we got going not to the ground 1 state, but going past the ground 1 state to the ground 0 state. So that's why it's an increased energy and increased frequency. Okay? Did that answer your question?
So there's the frequency, which is nu, right? The Greek letter that looks like a V. But then is that subscript like, is that frequency and then a
Oh,
subscript frequency?
this V right here is that the change in energy difference. So this is the frequency V. That's the frequency of visible, not visible. That's the frequency V, which is the change in difference in energy between the ground state 1 and ground state 0, which correlates to a vibrational frequency in the IR.
Okay, that makes sense. Thank you.
Okay. It's essentially the source wavelength with plus or minus subtracted value. Yeah, and that delta value can be directly correlated to a wavelength in IR. So imagine that the plus here or the minus here is what you're actually going to see. In fact, most of the time, because the Stokes is more intense, usually they just do everything via the Stokes scanner. Okay, so if we're looking for change in polarizability, we can even have the same molecule be very active in IR and not be active in Raman, and vice versa, by the different vibration that's happening. So for example, in the symmetrical stretch where both oxygens and CO2 are stretching out at the exact same intensity at the exact same time, that's equal and opposite. Therefore, there's no change in the dipole. And that means it's IR inactive. We don't see it, okay? However, if it's an unsymmetrical vibration, one oxygen moving in, one oxygen moving in the same direction here, we do have a change in dipole, and therefore it is IR active here, okay? So that means things that are IR active tend to not be Raman active. So things that are IR active tend to be Raman inactive, because all we're looking for is that polarization change, not that dipole change. So that means that things that are changing the dipole become Raman inactive. Okay, so that's why we can look at an IR spectrum and a Raman spectrum, and we're going to see peaks show up in different places, but they're associated with those same functional groups. Just one is going to be in a symmetrical stretch or compression, and one is a non-symmetrical stretch or compression. Okay, so in general, IR tends to emphasize any polar group, any group with a lot of different motion. Raman tends to emphasize aromatic and carbon-carbon double bonds, carbon-carbon single bonds. Because remember, the carbon-carbon bond is a nonpolar bond, and just vibrating a nonpolar bond back and forth does not change the dipole, but it does change the polarizability. So we can start to see carbon-carbon triple bonds, carbon-carbon double bonds, and carbon-carbon single bonds show up very strongly in Raman, where we wouldn't see them, or they'd be kind of in the baseline in IR. Now, there's other bending frequencies that do change the dipole moment, and the bending frequencies that change dipole moment are IR active, but are not Raman active, okay? So that gives us a kind of a complementary technique. When you do one, you can do the other, and you are actually supporting information about, oh, that's the symmetrical vibration, and that's where it should be, and that's the non-symmetrical, and that's where it should be. And one will be in IR, and one will be in Raman. The other cool thing is that because really polar stuff, or things with strong changing dipoles, cannot be seen, we can actually do Raman in solutions, okay? And again, most of the time our excitation is using visible light, so that we can use a system where things that would not normally be very easy to prep for IR can be prepped for IR. Like a protein in solution, you don't have to get a solid for it. You can actually do the stuff in solution as you go. Okay, so let's, yes.
The fingerprint that we can't read in IR, we can read it in REM spectroscopy.
Well, the fingerprint region is all of those unsymmetrical stretches for carbon, oxygen, oxygen, all those single bonds between all the different elements, except for hydrogen, are all going to be down there. And well, they're not, it could be a lot of them, there could be, you know, a wags or wiggles or rocking motions. Those all show up in different places in the IR, in the fingerprint region of the IR. If we do a Raman on it, what we're going to see is those symmetrical elongations of bonds that just change, like maybe the bond length, but not the dipole moment. And so we will see stuff in that region as well. But we might see peaks in the Raman we don't see in the IR. And they'll be associated with any kind of vibration that does not create a change in dipole. Okay. So let's see a comparison for that right here. So if we look at this first one right here, I'm going to blow it up and I'm going to use red here so we can tell the difference. Okay, so in the IR, we see what we expect to see. We have our hydrogens for our benzene and our methyl groups show up here. And then we have, of course, our benzene, you know, this is our carbon-carbon double bond region here. And then we have a couple of these carbon-carbon single bonds down here in the fingerprint region, okay? But in Raman, what we're seeing here is we're seeing these are symmetrical stretches of those CHs, okay? So these are so in these up here, these are like wags and stretches and things going back and forth like this that change the dipole. This would be more of like all three hydrogens stretching out at the exact same time and coming back at the same time, so it doesn't change the polarizability. So that doesn't happen very often, and so we do get a Raman peak, but it's not very strong. Now, there are also some peaks here associated with our carbon-carbon double bonds. Okay, things that aren't like twisting out of plane or something like that. It's a very symmetrical carbon-carbon double bond stretch, and so we see those in the Raman quite well. But notice we see a couple other peaks we don't really see very in the other system here. We don't see these. All of these are going to be carbon-carbon single carbon-carbon symmetrical stretches. Probably the first one's going to be due to the CH3s moving in and out from that benzene ring. These are probably going to be CH bands as well, in moving in a different way around these methyl groups. And because they're not changing dipole, we don't see them in the IR. So we can get some diagnostic work for it. For example, let's say we probably can tell the difference between the different isomers of this trimethyl benzene group because it is symmetrical here. And so we're going to have bonds that we're going to have peaks in the Raman spectrum with the more symmetrical molecule than if we had it in a different isomer that wasn't as symmetrical. Raman favors symmetry. Raman favors linear molecules. Raman favors those kind of things. So we should be able to tell the difference between symmetrical and non-symmetrical isomers using the Raman spectroscopy. Okay, let's run through another one here. Again, we're probably seeing carbon-hydrogen symmetrical stretches here. We see a lot of the same bands we see in the IR here. And so they must have a symmetrical and non-symmetrical stretching mode. But then we have a couple bands that we don't see in the Raman in the IR at all that show up. And those are most likely associated. Again, all of these are associated with carbon-carbon double bond symmetrical stretching. And these are probably due more toward the carbon-carbon single bond symmetrical stretching on these. And these are just the different types of hydrogens we have here. Okay. So do you see how you're adding additional information with this system? And sample prep is so much easier. They have now Raman machines that you just basically put it in a capillary tube, hit it with light, and you get your spectrum out almost, just after 64 scans, just like you would normally do. They even have handheld devices. You just stick it in a bag and you can get a Raman spectrum and then compare it to your library. And you can see if your bag of grain is contaminated because the Raman spectra will change that much by having something that is not there, should not be there. Okay, questions on how to use and the concept of how Raman spectroscopy works. Okay. So now I'm going to show you a few instruments and then a few applications so that we can get kind of an idea of how to use it in our research as we move forward. So the basic idea is we have to have a very intense light source. We have to focus that light source on our sample. And like I said, our sample can be in a tube. Sometimes it's a test tube, and so it can be anything here. And then you have a reflector here. Notice that we're not, this is our incident beam. Our incident beam is going this way. We're not measuring the absorption through the system at all, okay? But our detectors typically are at right angles, either a 90 degree or 120, 270 degree. Sometimes you can actually put the detector back here. You're looking for 180 degree reflection here. And those are different techniques, but the most common is this 90 degree off angle so that your incident light does not flood your spectrophotometer's light detector, okay? And like I said before, we typically use visible light as the intense source because it's really easy to use quartz. I'm sorry, it's really easy. Visible lasers are really easy to work with because you can see them. They're not as hard to work with as UV or IR lasers because when you can't see them, you get injured with them. And also we can also use regular optics. We don't have to use like silver iodide windows or anything like that, like you need for an IR or other systems. We can use just glass and regular systems. And we can use UV vis type detectors because remember, we're looking for that change in frequency of our incident light, not necessarily an IR signature out of it. So we're using really nice, simple detectors. So imagine just taking apart your UV vis and then making the source at a right angle and sticking a laser on it. Okay. So the cool thing is that you can actually use it for quantitative reactions because just like IR having that fingerprint region, Raman is very sensitive to things that are not symmetrical, don't have that bond polarizability. For example, Raman you can even use to like measure water content sometimes because that will change the polarizability of some of the bonds. Okay. And so you have to be careful of that. You can actually do samples with aqueous things, which means you can do Raman spectroscopy on living cells. And that's one of the applications we have. And so we can use it in medical, in biological, it's used a lot in food, it's used a lot in quality control, okay? Because we're using visible light and we don't have to use all these fancy windows that are IR transparent, we actually can, it makes it easier to use, okay? Occasionally there's an interference with fluorescence. For example, if you have a dye that's absorbing and fluorescing in that wavelength, the fluorescence actually emits out 360 degrees C, might pick it up, but it's not that much. The only reason that the quantitative information a little more expensive is the fact that you're using a laser system. And lasers have actually come down in price so that they're actually getting pretty reasonable. A handheld version you stick in a bag of wheat costs about $3,000 now. Okay. Okay. So this allows detection all the way down to about 50 wave numbers. It's that entire IR range, 4,000 wave numbers down to 50 wave numbers, because again, we don't have to worry about having a light source to do the absorption. And we can also take into account surface plasmon effects. And I'll talk about that when I talk about surface enhanced Raman spectroscopy. Surface plasmons are where you can isolate energy on the surface of metal particles, and therefore they can enhance the Raman signature. And that, yeah, I helped in that. So cool thing is most of them, you know, they look just kind of like an IR with a laser. Most of them have a safety shield, so because of the laser light. Most of them have very easy sample holders. Again, you can use test tubes or capillaries so that it's very easy sample prep. You don't have to purge it for CO2. You don't have to, you know, worry about water in your system. So it's really, really a durable system. But one of the more common ways to do it is actually hook it to a microscope. So especially when you're doing something like a biological system, what you'll end up doing is using a microscope so you can focus on different parts of the cells and pick up different amounts of energy. And if you scan the whole thing as you're doing that, you get this map of all the different certain Raman intensities as you go through the molecule or through the cell. And so you can actually say, oh, look, this is a lipid, high lipid area. This is a high protein area. This is mostly water, and or you can look for amino acids as well. So the microscope is actually really, really common. It's also good for inhomogeneous materials, so that you can say, oh, if you, let's say you had a rock and it had different regions in it, you could take it on this and go, oh, look, this is the feldspar, this is the quartz, this is the chert or whatever. And so you can actually determine the different chemical compositions within a granite, let's say, because you can focus in on each individual section. Okay, so one application is a biological application. This particular one is that looking for disulfide bridges in proteins. So you may know that disulfide bridges are how proteins maintain their structure. So when they're made or when they're put through their enzyme, they'll fold to a certain place, and then the cysteine on each side will bond and hold it in place. So it's actually a full sulfur-sulfur bond, that disulfide bond, holding this protein in place. So you can easily denature a protein by breaking those disulfide bonds. Okay, so the bad thing about that is that you have all these other functional groups in there, amides and alcohols and amines and carboxylic acids. So they're very noisy if you look at them by IR. And that disulfide bond would be very, very weak. It does have some unsymmetrical stretches, but it's very strong as a symmetrical stretch. So that makes it a good target for Raman spectroscopy, because we have that sulfur-sulfur bond. Okay, and so what that would look like is, let's say you're trying to denature a protein. And so you started with your spectrum and it looked something like this. And notice this is arbitrary relative intensity units. In this particular case, they used a deconvolution software to figure out where the peaks of the different peaks under that curve. The actual red is the curve that you would actually see. And then there's some software it'll mathematically fit peaks and curves underneath that. And what we see here is this entire section here is the different types of disulfide bonds. And you can see a great diminishing of those disulfide bonds as you go into the system. And so it looks like maybe this is an overlapping peak, but didn't change. This is our major disulfide bond, and it did significantly change. And so we can actually watch this change. And you can also, there's a lot of amide bonds in there, and you can see it, the shifting of the amide bond as it moves around as well. Okay, so that's one biological application for it, is in protein chemistry. Another one is for tissue analysis. You can actually take some systems and actually do it on live humans, where you look for different little systems here. Or you can do it with, you can send it off for a pathological test on it. And by looking at normal tissue and seeing what kind of amino acids, et cetera, are in there, and which kind of things that you should see or shouldn't see, you can actually compare normal tissue to things that are just abnormal growth and things that are actually cancerous, because they have different chemical tags on them. And so you can actually go in, take a slice, take your Raman microscope, and look for these emissions from the system, and you can see that and compare that to normal tissue, and you can quantify, well, not quantify, you can successfully say, oh, this is definitely a cancerous tissue. And if you have enough of it, you can go around and figure, okay, it's located to this very small area. And so that we can actually find all the margins on this cancer cancerous area. So that'd be kind of cool if you were going to go into the biomedical field as well. Okay. Again, you can do live cell analysis because water does not impede Raman because we're using visible light again. And the stuff that's coming out is visible light, just shifted slightly. And in these cases here, what they've done is they've mapped the area here by going several sections over and several sections down, and then looked for the intensity and wavelength changes. And so in this particular one right here, we actually are looking at the fat content in the cell that we have in this particular one. We're seeing that our fat content is highlighted in green right here. And so there's a large group, a lot of energy storage in the cell right here because of that fat storage. Okay. And again, this would be done with one of the microscopes that has a scanning function, and it would go over the entire system and map it out. That takes a little bit longer. But now this is something I know a bit about. I've published on it a couple of times, and it's called surface enhanced Raman spectroscopy. So when you have small nanoparticles, specifically, we usually use gold and silver because they're nice and stable once you put stuff on the surface there. And you use the idea that if they're close together but not touching, that you can quantize or trap energy, specifically surface plasmon, in between the two cells. Now, so now you have this extra little burst of energy available to you with the surface plasma. And then the gold surface being very active because it'll absorb different analytes to it. We were using them specifically to look for explosives. And so we could take a 1 part per billion solution of TNT, and using this SIRS effect on these specially treated gold particles, I synthesized the gold particles and the surface treatment. We were able to get this enhanced emission from these, femtomolar explosive compositions. And they show up really well because, again, the intensity is all about that absorption, and that surface plasmon gave additional energy. It actually, instead of being 0.001% of the signal, it actually boosted it up to about 1%, 0.01%. So it's a 10 time to 100 time boost in signal output from this surface enhanced Raman spectroscopy. And so, but that was a really cool project. Okay. I actually just learned this today. I didn't know this before, but if you use polarized laser light, you can actually see whether or not a compound is chiral or not. So by analyzing the light coming through, if the light has rotated or not rotated, you can actually see that a particular compound has optical activity. Looks like a very hard way to do something we can already do in just solution as is, but I thought that was really interesting. I thought you might think that's kind of cool. So Matt, and so, yeah. And that's my last slide on Raman spectroscopy. So again, it's just an introduction. The basics are excite with a laser and then look for your Raman active sections in the IR. Okay, questions?
So like in IR, they have, we have 5 regions that we see that if these regions are, then we can find these functional groups sort of thing. It's, are these kind of region exist in the Raman?
They are the exact same regions. So
okay.
if your compound is IR active, let's say the OH bond, if your OH bond is IR active, okay, it's going to show up in that first band region, right? If there happens to be a non-symmetrical, just a polarizing absorption available to it or a polarizing vibration available to it, you will see it in Raman in that exact same place. Okay, so the bands are the same, the locations are the same, the functional groups are the same. It's just looking at a specific vibration that is not changing the dipole moment of your molecule. So that's why I said when we looked at these right here, I said, oh, this one must be part of a carbon-carbon double bond, asymmetric stretch. This must be part of a carbon-carbon double bond, because we learned all of these are carbon-carbon double bonds found in benzene system, and because there's all the different varieties there. So these just happens to be ones that don't change the dipole moment. All the ones up here are the ones that do change the dipole moment, but it's the exact same functional group. Okay, other questions? Okay, so again, I wanted you to see that this is something that's available to you here at the university and as you move on with your research. I haven't worked in it in 14 years now, so which is why I'm a little rusty on it, but so no, I guess it's been 10. But I wanted to make sure that you saw that there's value in this particular form of spectroscopy as well. And the fact that it complements IR, it uses the same wavelengths and bands, it's in functional group identification. It's a good little tool to have in your toolbox. Okay, so that's all I have on Raman spectroscopy. So the rest of this, we can just have question and answer about the exam. Again, we're focusing on proton NMR, carbon NMR, and 2D NMR. And we'll have one set of questions on UV vis, but no Raman. Okay. And there, as always, there'll be one solve the entire molecule from your IR, mass spec, proton, and carbon NMR. So you have to build every little fragment from all the different pieces of information you're getting.
So do we like, what about the heteronuclear ones?
The
heteronuclear ones?
Hetero,
sorry. The nitrogen, phosphorus, silicon?
Oh, no, the other night, no other nuclei. That's why I did it as an exercise so that you would get experience in it. But number one, it's really hard to find them in the literature. It took forever to find just those examples. And our book only has a couple examples too, and you would have those examples because you have the book. So it was very hard to find even those. So I chose to use them just in that because we hadn't actually worked out a problem with it. So I thought working out those problems together was a better way to get you to learn it without having to stress about it.
So same goes with the final exams. They will know.
Yeah, the final exam is going to be where, in fact, I think every question is going to be that one-page thing. Here's the IR, here's the mass spec, here's the proton NMR, here's the carbon NMR, and if it's a really complex compound, I'm going to give you the 2D NMR. So the final exam is all problem solving. Okay. So this one's going to have one set of questions on UV vis. This is going to have probably identify every proton and carbon based on 2D NMR, and then one problem where you solve the whole thing, and then another couple that are just proton and carbon NMR issues. All right. Well, if you have to run off and go do something else, go ahead. If you want to stay behind and ask a few questions, go ahead. I'm going to go ahead and stop recording. So that's at least